Today's programme for the course Biology, Form and Function looks at the way in which biochemistry and electron microscopy have been brought together to unravel the relationship between structure and function in the rod cell in the retina in the eye.
When I entered this darkened cinema from a bright exterior, I actually suffered momentary blindness. But the eye's a remarkable structure. It's got a wide range of sensitivity. In fact, I'm already beginning to pick out the surroundings in this room. But I'm picking them out in very little colour, very little detail. And what's more, I'm not going to see any colour until the lights come on. Now, this suggests that there are two types of cells, two types of sensory cells operating in the eye. The one which is operating in a bright light intensity, giving colour vision, and the other one operating in dim light intensities, like this room, giving black and white vision. But in order to understand this, we've got to look at the anatomy of the eye, and in particular, the anatomy of the retina. Using a video camera attached to an ophthalmoscope, we see the retina laid out like a film on the back of the eye. You can actually make out the blood vessels running through it. Looking in more detail, we see that the retina is made up of a series of different layers, but it's this layer here which has the light receptor cells. These are the rods. They're called rods because of their shape. And these here are the cones. But which is responsible for colour vision and which is responsible for monochrome vision? A clue comes from looking at the retina of a nocturnal animal. They have a very high proportion of rods. So it looks as though rods are the cells which allow vision in dim light intensities. But how can these rod cells produce visual images in the brain? Light certainly affects these cells in some way, producing an electrical signal in the optic nerve fibre. But how? The real question then is, what is the link between light and the electrical impulse in the nerve fibres connected to the sensory cells? Dr. Colin Kemp, Institute of Ophthalmology.
We've just seen the colour changes that take place when a fully dark-adapted retina is exposed to room lights. Initially, the retina was bright red, and rapidly, under the room lights, the colour faded. That fading is the clue to the link between the absorption of light by the retina and the generation of the neural signal from the retina. The red colour is due to the presence of the visual pigment, rhodopsin. Rhodopsin consists of a protein, opsin, covalently bound to a specific derivative of vitamin A. When it absorbs light, rhodopsin undergoes a series of reactions culminating in the dissociation of the protein, opsin, from the vitamin A. As a result of one of those reactions, the receptor signals the absorption of the light. If we want to look at these changes in more detail, we can do so quantitatively by placing the retina in a spectrophotometer. I've mounted a retina inside this spectrophotometer on this microscope stage. A measuring beam passes through the retina. It's a very dim measuring beam so that it produces virtually no bleaching of the pigment, and it falls onto a photomultiplier. The output of the photomultiplier is then displayed on this XY recorder. As the wavelength changes, so the recorder moves this way. So, on the left, we have ultraviolet, then violet, then blue, blue-green, yellow, and over here, red. A characteristic of the visual pigment of the rods, rhodopsin, is that it absorbs maximally in the region of 500, that is, about here. Now let's see what happens if we expose the retina to a very intense, brief flash of light. As you can see, there's been a really quite dramatic change in the spectral properties. We've lost a lot of the absorbance here in the blue-green at 500 nanometers, and we've gained correspondingly a lot of absorbance in the ultraviolet at about 380 nanometers. What has happened is that we've bleached a large amount of the rhodopsin and formed an equivalent amount of a photoproduct. Almost immediately after the absorption of the light by the pigment, a small conformational change takes place within the vitamin A derivative part of the molecule. This change is accompanied by a very large conformational reorganization of the opsin part of the molecule and is accompanied by the bleaching, the decolorization.
So in some way, this conformational change in rhodopsin is related to changes in electrical energy in the optic nerve. So now let's look at the anatomy of the rod cell and examine where its rhodopsin is located. Dr. John Marshall, Institute of Ophthalmology.
You have about 120 million rod cells in each eye. And to give you some idea of scale here, if the whole eye were magnified to the same extent as this model, it would be 3/4 of a mile in diameter. As you can see, the rod itself is divided into 4 major regions and a series of interconnecting zones. The light-sensitive portion of the rod is referred to as the outer segment. Outer means is pointed towards the outside of the eye. As we travel up the rod towards the inside of the eye, that is the lens and vitreous, we reach a portion called the inner segment, this conical zone here. Further in is a bulbous nuclear zone, and then finally, at the top of the cell, a synaptic region or rod pedicle.
Looking inside the cell, the outer segment contains these characteristic stacks of disc-like structures. At the end of the outer segment, there's a thin junctional region joining the inner and outer segments. Mitochondria are particularly abundant just above this region. The nucleus is sited deeper in the cell. And finally, at the opposite end of the cell is the synaptic region connecting to the retinal nerves. Comparing this model with EM photographs, we see the discs are membrane-bound. They float freely in the cell's cytoplasm. A connecting cilium is usually found here. Finally, the synaptic region is filled with vesicles containing neurotransmitter.
Recent work relating structure to function in rod cells has shown that the inner and outer segment in the cell have a constant dynamic interrelationship. Using radioactively labeled amino acids, if these are presented to rod cells, they're initially taken up in the innermost portion of the inner segment, and label is found associated with the ribosomes in this region. A little later in time, the grain moves from the ribosomes to the Golgi complex and then diffuses through the mitochondrial portion of the inner segment until it finally forms a band of radioactive label at the junction of the inner and outer segments. This band moves down the outer segment as a band, a discrete band of radioactive label, until it finally reaches the tip of the rod photoreceptor cell. After a further period of time, the label is seen within the cytoplasm of the pigment epithelium in large secondary lysosomes or phagosomes. This work is interpreted as meaning that the discs in the outer segment are constantly being renewed. Now, further work has shown that the major portion of the amino acids involved in this turnover are incorporated into the protein portion of the rhodopsin molecule. So not only discs are being renewed, but rhodopsin's being renewed. And clearly, the labelled rhodopsin is trapped within the disc.
The rod cell has a long, thin structure. The discs and the synaptic region are some considerable distance apart. So how can this change in membrane-bound rhodopsin within the discs be related to changes in the synaptic region of the rod cell? Evidence suggests that ion movements are involved. Dr. Jeff Owen, Cambridge University.
In darkness, the inside of the rod is more negative than the outside by between 30 and 40 millivolts. The effect of light is to make the inside even more negative. That is, the rod hyperpolarizes. Now, if we increase the light intensity, the size of this hyperpolarization increases also. In other words, the size of the electrical response is related to the light intensity. How does this light response come about? In darkness, the membrane potential of the rod lies roughly halfway between the equilibrium potentials of potassium and sodium. And this suggests that as well as being permeable to potassium, the membrane is also highly permeable to sodium. Now, we can show that this is true if we bathe the retina in a saline from which the sodium has been removed. Here, initially in normal saline, when we flash a light, we elicit a strong light response. At this point, we change to a saline from which the sodium has been removed. The membrane rapidly hyperpolarizes by a further 40 millivolts. And now, when we flash the light, we elicit no light response. When we replace the sodium, there is a rapid repolarization of the membrane to its original dark level, and once more we can elicit a light response. Taking this in conjunction with the results of several other experiments, a picture of the rod has emerged in which sodium flows into the cell across the outer segment membrane, which is highly permeable to sodium. The concentration of sodium inside the cell is kept constant by ion pumps located in the inner segment, which pump the sodium out again. What this means is that in darkness, a loop of positive current carried by sodium flows around the cell. Not surprisingly, this is called the dark current. Now, at the same time, in darkness, synaptic transmitter leaks at a constant rate from the synaptic terminal of the rod. The effect of light is to reduce the sodium permeability of the outer segment membrane. This interrupts the current loop, reduces the current, and causes the cell to hyperpolarize. At the same time, the rate of release of synaptic transmitter from the synaptic terminal is also reduced. And so the absorption of light by the rod is signaled to the rest of the visual system.
Variations in equilibrium potentials suggest changes in ionic permeability. Current changes provide a direct link. Dr. Trevor Lamb, Cambridge University.
Now, in order to measure this current, we suck the light-sensitive outer segment into a tiny glass pipette. Normally, the current flows in a local loop like this. But if a pipette is slipped over the outer segment, then the current can no longer flow in this local loop, but is forced to flow in the longer loop through our pipette. The saline solution, which is bathing the lower part of the cell, can be made separate from this saline solution inside the pipette. And in this case, the current will no longer flow in this loop. But if we connect a wire from this solution round to the solution inside the pipette, then once again the current can flow in this loop. And now all we have to do is connect a sensitive current detector into this wire, and we're able to measure the current which flows into the outer segment. We do this kind of experiment under infrared illumination. The pieces of retina are in this glass chamber, and the pipette comes in from the front and then is connected out to the suction and also back to the electronics. In order to be able to see these photoreceptors without exposing them to light, we use this infrared sensitive TV camera connected to the microscope. And in order to position the pipette on the screen, I simply adjust these knobs. And with the front of the screen of the cage down, I can position the pipette as necessary. Now I'll move this piece of retina up towards the pipette. Focus on an outer segment. Focus the pipette. Well, now that the cell is fully into the pipette, I can turn on a light stimulus which fully covers the outer segment. And also I can present it in the form of brief flashes. These are brief flashes once every second. Now, so far all of this has been done using infrared light, but we can now set up the optical stimulator with visible light. And the current that is recorded by the pipette is displayed on the chart recorder and on the oscilloscope. And now I can give a bright flash of light. The response to this bright flash is a rapid upward transient in current, and after a few seconds it recovers, and then a second identical flash gives a very similar response. This level at the top of the screen corresponds to zero current, and this level down here corresponds to an inward current into the outer segment. So during the light response, the inward current has been reduced. And this is because the cell membrane has become less permeable to sodium ions. The amplitude of the rod's response is graded with light intensity. With a dim flash, there's a very small response, which is barely detectable above the noise. And then with a brighter flash, there's a larger response, and so on as the flash intensity is made brighter. With bright intensities, the response saturates at a level corresponding to complete suppression of the inward current, and still brighter flashes give no additional response amplitude, but simply prolong the response. So over quite a wide range, the light intensity is signaled by the amplitude of the cell's response. The experiments with dim lights, these small responses here, show that the rod is extremely sensitive to light and can actually detect single photons, the elementary units or particles of which light is composed. This figure shows a cell's response to a long series of identical, very dim flashes, and is recorded at high amplification and on a slow time scale. And each of these 4 traces represents 10 responses to these identical, very dim flashes. And it's clear that the size of the response varies from trial to trial. Sometimes there's been no response at all, sometimes there's been a small response, and occasionally there's been a larger response. Well, this is because at these very dim intensities, the rod is hit sometimes by no photons, sometimes by 1 photon, and occasionally by 2. Well, it's this phenomenal sensitivity of the rods that enables us to see even under conditions of starlight. But what's perhaps equally amazing is the rod's ability to continue functioning up to light levels thousands of times higher than this. We can also ask what particular color of light the photoreceptor prefers. And with the optical stimulator, I can vary the wavelength or color of the incident light. By rotating this disc, I can vary the color of the stimulus beam. This is 700 nanometers red, 600 nanometers yellow-orange, 500 nanometers green, and 450 nanometers blue. A bright blue light, 450 nanometers, gives this response. But if we present the same number of photons at 500 nanometers, we get a larger response. And then at 600 nanometers, yellow-orange, the response is again smaller. And at 700 nanometers, deep red, there's virtually no detectable response. So we can plot the cell's sensitivity as a function of the wavelength or color of the stimulating light. And these are the points we obtained. So the spectrum of the sensitivity of the photoreceptors looks like this. And this is exactly the same curve as was obtained for the absorption spectrum of the pigment rhodopsin, with a peak at 500 nanometers. So the initial conformation change in rhodopsin must be linked in some way to the electrical responses that we record from the rod. So what does this mean in terms of the mechanism of the rod's response? Most people working on photoreceptors believe that these linking events involve a change in the concentration of a substance inside the cell, and that this internal transmitter substance affects the sodium permeability of the plasma membrane. But just exactly what this substance is somewhat controversial.
Unilluminated discs contain high concentrations of calcium ions, and it's these that may be involved in transduction. Light interacts with rhodopsin, causing a conformational change, and this increases disc membrane permeability. Calcium now leaks out down its concentration gradient. These calcium ions then interact with sodium channels, causing them to close, so switching off the sodium current. Now, although this model is simple and convincing, the link in reality may be more complex. Changes also occur in cytoplasmic levels of cyclic AMP and cyclic GMP, indicating enzymic involvement. Whatever the nature of the link, its function is one of amplification.